Hello, I'm Dr. Sagar Lonial from the Winship Cancer Institute of Emory University in Atlanta, Georgia. And on behalf of my colleagues, I'm presenting the Iberdomide in combination with dexamethasone in patients with relapsed refractory multiple myeloma, results from the dose expansion phase of the CC-220-MM-001 trial. Again, I'd like to thank my colleagues for the opportunity to present this data on their behalf. So as you know, Iberdomide is a novel cereblon E3 ligase modulator, or CELMoD, and is being developed as the next generation IMiD agent in myeloma. Preclinical data has demonstrated pretty significant synergy with DEX, CD38 antibodies, and proteasome inhibitors, and is able to overcome both lenalidomide and pomalidomide resistance. And what's unique actually about Iberdomide, different from those characteristics above, is that from a chemical perspective, it is just the S isomer of the compound, which actually may result in less adverse events, as it's known that the R isomers seem to be associated with the sedative side effects and fatigue associated with the IMiD class. The two cohorts I'm going to present are cohort D, which is the expansion phase of IBER DEX in the context of triple class refractory myeloma. And the second is cohort I, which is the IBER DEX combination in patients who have previously been exposed to BCMA-directed therapy. As you can see, the two cohorts are really very heavily pretreated, between 6 and 7 years from diagnosis. If you look at the percentage of patients that are both POM and LEN refractory, in cohort D, it was almost 85 to 95%. In the post-BCMA, it was 88 and 84%. And most importantly, if you look at the people that got prior BCMA, about 50% of them had gotten prior antibody drug conjugate as their exposure to BCMA. One of the unique features, I think, of Iberdomide is the adverse event profile. As you can see, there is some hematologic toxicity, not really unique or new to this novel mechanism of action, but certainly some of the fatigue, diarrhea, constipation, and rash, particularly associated in grade 3 and grade 4, are pretty low in this. And in fact, if you look at cohort I, it's even lower. So these numbers really are quite consistent with what we've seen in the early phases studies of Iberdomide and dexamethasone. So here's the overall response rate, 26.2% for the IBER DEX expansion cohort and 25% in the BCMA exposed. I will note there were a couple of patients who were down here in the MR or stable disease category that unfortunately had to come off because of the development of COVID. And so they didn't have a chance to deepen their response, but certainly there is a very good response overall. When you look at duration of response and time to response, time was really quite quick. Median PFS was about 3 months. Median duration of response was about 7 months in aggregate. And if you look at overall survival, again, what I think you'll see is overall survival for the responders is really much longer than you'd expect for a similarly heavily pretreated triple class refractory patient population overall, demonstrating not only are patients getting clinical benefit, but that's translating into an improvement in overall survival as well. From a PD perspective, you can see we are hitting the target. This is the known putative downstream target of cereblon. And equally as importantly, what you can see is we're activating NK cells and T cells, which again speaks to the CELMoD mechanism of action. So in conclusion, in heavily pretreated triple class refractory patients, IBER DEX demonstrated clinically meaningful and significant responses, especially in patients with triple class refractory and BCMA resistant myeloma. The treatment was well tolerated with very few grade 3, grade 4 non-heme adverse events. And again, as a clinical metric of that, the relative dose intensity was over 88% of IBER, with very few dose reductions or modifications needed to keep people on treatment. Overall, the health-related quality of life was maintained. I didn't show that data, but it certainly was maintained. We're hitting the target and stimulating the immune system overall. And these results are leading to a phase 3 trial combining IBER with proteasome inhibitors as well as CD38 antibodies, which we hope will ultimately lead to approval.